All right. Good afternoon, everyone. My name is Rupak Karan, and I'm a PhD candidate in the Department of Plant and Soil Science. I'm working with Dr. Wanjun Guo in the Crop Ecophysiology and Precision Agriculture Lab. And I'm also serving as the president of PSS Graduate Council. So, all esteemed guests and distinguished colleagues, it is my honor to introduce today's speaker for the PSS Spring Seminar 2024, Dr. Terry Wheeler. Dr. Wheeler is a professor of soil-borne diseases of cotton and peanut within the Department of Plant Pathology and Microbiology at Texas A&M AgriLife Research, Lubbock, and an adjunct faculty member in the Department of Plant and Soil Science at Texas Tech. Her research in cotton has focused on the management of nematode-related problems, Verticillium wilt, bacterial blight, seedling diseases, and Fusarium wilt. She grew up on a small farm in upstate New York and completed a BS degree in interdisciplinary studies in Worcester Polytechnic Institute, which included 1 year spending at the Cornell University. And then she spent 2.5 years working in Honduras as a plant pathologist in the Peace Corps. She earned her master's degree, came from the Texas A&M University, and her PhD at North Carolina State University, with both degrees being in the plant pathology with an emphasis on nematology. She then worked on Verticillium wilt of potato and soybean cyst nematode for Ohio State University as a research associate before taking the faculty position at Texas A&M. Please help us welcome Dr. Terry Wheeler to share her insights with us today. Thank you.
Okay, so I'm a plant pathologist. I've been in love with plant pathology ever since I was a sophomore in college. And so that's when I discovered the field and have been taken by it ever since. I was particularly interested in nematodes and soil-borne fungi. And Fusarium wilt is a disease that has both as part of the disease complex. So the fungal part of the complex is Fusarium oxysporum, Forma specialis, vasinfectum, FOV for short. And the types that we tend to have out here that cause wilt symptoms are race 1 or race 2. But it is a complex, and we don't typically get the problem with the disease unless root knot nematode is also present. And so what happens is the farmer plants the field, the cotton comes up really nicely, has a good stand, and then maybe 30 to 45 days after planting, the plants start to do this. They start to die, and they'll go down rapidly. Usually by the 4th of July, his field will look like that. And this is a case in Western Gaines County where the farmer just turned the water off after that. He just said, I'm done. So that's really a catastrophic disease. It doesn't happen every year or for every field. It is a bit sporadic to actually be catastrophic, but it's one of those diseases where the entire field can get wiped out, the entire field. So it is catastrophic, and it occurs late enough that it's too late to replant. And so it does sort of fulfill my definition of a catastrophic disease. Now, the root knot nematode is part of it. In the upper left side, you can see the galls for root knot nematode. One of the diagnostic symptoms is this root discoloration. So if you slit the root, you will see right in the center of the root, you will see a discoloration. And that is an unusual symptom. Most fungal diseases do not cause that symptom. So that's semi-diagnostic when you see that. So here we go. We have the fungus here growing on a plate. This is Fusarium. This is the root knot nematode, the second stage juvenile, and the types of galling that you get with root knot you can see on this picture. So it gets its name because it sends a message to the plant, and it begins to form this gall around the nematode. And it does that, it does not inject DNA like Agrobacterium does to form a gall. It's a little bit further downstream, but it is sending messages to the plant where the plant then starts forming this tremendous amount of cell division and cell elongation to form these galls around the nematodes. So if you look at the disease cycle, here we have some dark colored chlamydia spores. That's the fungus overwintering structure. It can also overwinter as mycelia that's embedded in plant debris. And you get a plant growing in the spring. Here's your little root knot nematode here. So it infects, and when it infects, it creates a little bit of wounding, but more importantly, you get a root exudates coming out. You get leakage of material. And so it's very attractive for the Fusarium then to take advantage of that. It colonizes the outside of the root, but then it gets internally. It's able to actually enter the center part of the root. And most fungi don't colonize internally very well, but this is able to do internally, and then eventually it moves upward into the stem in the above part of the plant. And you see the spores up there, the tiny microconidia, and then the more boat-shaped macroconidia. Those are the spore structures that will flow upward in the plant. And so once that happens, if it's not too bad of an infection, the entire plant can be colonized, including the seed. The seed can also be colonized. And then when the plant dies at the end of the growing season and it is laying on the ground, that inoculum is being dispersed in the soil. So you're not going to get wind blowing inoculum. It's not an airborne disease. Its spread is fairly slow unless you count the seed, which could be then spreading to new fields. But it does survive really well. Now, the plant, if it dies, then you have a smaller amount of plant that's actually in the soil. So you actually get more inoculum if a plant's not that bad that it dies prematurely. If it actually survives all season, it actually builds up more inoculum. But if it dies early, then that plant material can be a food source for the fungus even after it dies. So this fungus has a wide host range. It doesn't cause disease on a wide host range, but it can survive and colonize a wide host range. It's an excellent saprophyte, so it lives on dead material as well. It can colonize all parts of the plant, including the seed, and it has this chlamydia spore, which allows it to survive for years. Okay, so with FOV, or Fusarium oxysporum vasinfectum, we have several different races. And for a lot of plant diseases, if you say a fungus has a different race, it's because it has overcome a resistance gene in a variety, and it's sort of mutated, and it has you have to come up with another resistant gene then for that subpopulation that has overcome a resistant gene. Well, that's not the case in FOV. The race situation is really different hosts. It has different hosts. So the difference between, for example, race 1 and 2, they're exactly the same on cotton. But if you were growing tobacco, some species of tobaccos would be a host and some would not, depending on which race you have. So it's a little counterintuitive with FOV. So we have what we call the vascular competent, and those require a root knot nematode. And then we have the root rotting type. So the vascular competent is what spreads upwards in the stem and gets above ground. And race 1 and 2, which is common out here in the Southern High Plains, can do that. Race 8 can do that. I'm not sure if we even have race 8 in Texas. We don't have it out here that I'm aware of. Race 3 and 5 are the root rotting type. 3 and 5 are difficult to tell apart. So that's why I put them together. We have race 3 out here as well, but I've never been able to isolate it from the above ground stem part. It's only from the roots. And then race 4 and 7 are, again, you can't really distinguish them easily. We do have race 4 in California in the US. It's in far west Texas. It's in China and India, but it's got a fairly limited distribution. It's extremely aggressive. And then Australia has a race as well that is a root rotting type. And the way we do races is there's 3 nuclear genes that are amplified, or at least part of the genes are amplified. The translation elongation factor, the phosphate permease-like proteins, and the beta tubulin. And by using those 3 genes, you can then determine whether you're race 1, race 2, race 8, etc. If you have race 4, there's been some faux sequences that have an insertion called this T-fold. And so there's an additional type of subdivision within race 4, depending on whether you have this insertion or not, and whether that insertion has some mutations in it. All right, so we're going to look at the interaction of the nematode and the fungus. And the point I'm making on this slide is it's density dependent. That means the more you have, the worse your disease. Okay, density dependent. So this is some work done by Dept in California. And he had a field with both the nematode and the fungus, and he artificially manipulated things, like he fumigated some plots, or he used solarization to kill organisms in plots to try to create different densities. So in 1982, he had a high density of root knot and a high density of FOV, and he had a very low density, and he got no wilt where he had the low density, and he had 90% wilt incidence with the high density. In 1983, he was able to get 3 different levels going, and you can see the incidence of wilt was very, very high where he had a high density, and not much of a problem where he had a medium or low density. And then in 1984, you can see really the medium and the high both had a considerable problem. So density is important with this disease. Back in 2003, I started getting a lot of calls about Fusarium wilt, catastrophic Fusarium wilt. I hadn't had many calls prior to that. I'd had a few, but they were very unusual. So if you look at the acres, the planting acres of FiberMax, you can see in 2000, Texas had only a few acres. And by 2005, we had over 50% of the acres were being planted with FiberMax varieties. So 2003 is when I started getting these calls about catastrophic Fusarium wilt loss. Well, every single one of those fields had been planting these new FiberMax varieties for 2 or 3 years. Every single one of those fields. So something was going on with those new FiberMax varieties. They were developed in Australia, and at that time, Australia did not have Fusarium wilt, and they did not have root knot nematode. So they were developed in an environment where the disease did not exist. We brought it to the US and we started planting them because they were very well adapted to this region. And bam, we started having Fusarium wilt problems. So this is a test that I did in 2004 in one of these fields that had catastrophic losses due to Fusarium wilt. On this graph, I have the mortality. So mortality is when you get a stand initially and then the plants die. So the percent of plants that die, that's mortality. So the red bar is where I had these new FiberMax varieties, conventional, the ones that were straight from Australia. The black bar were the FiberMax varieties that were transgenic. So they would have had an additional cross to bring in the transgenic trait. And then the blue bar is every other variety that was susceptible to root knot nematode. It was, Delta pine and all the various types that were around back then. And then this odd looking yellow bluish bar, that was, we had just come out with the new, with a few nematode resistant varieties. They were very new. It was the first time we really had nematode resistant varieties. And so you can see in 2004, going into one of these fields that was catastrophic the year before, well, it was catastrophic in 2004 as well. We averaged over 80% plant mortality, plants that came up, and then 80% died where we had the conventional FiberMax. But really all the other varieties had also just catastrophic losses. They were over 60% of mortality. So everything was just getting destroyed. Yes, the original FiberMax varieties were worse, but everything performed badly. Now, if you look at the root knot nematode densities in the soil, they weren't that high. For this region, they were over 1,000 for most of these variety groups, but that's not a particularly high number for out here. But you can see that the resistant varieties did have lower densities. So the resistance, the root knot resistance was working. And then if you look at the yield, the conventional FiberMax only made 600 pounds. The transgenic FiberMax doubled that at 1,200 pounds. And then all the susceptible varieties averaged about 1,000 pounds, and the root knot resistance was around 1,200 pounds as well. So lesson one, when a field is smoking hot, everything's going to die. It doesn't matter what we plant. The question is, how did it get so smoking hot? So here's 3 years that I spent at that field. So the field had a history of FiberMax 819 prior to 2004. I asked the grower to use one of these nematode resistant varieties after that. And so he did. He planted everything else in the field with a nematode resistant variety, except where our test was in 2004. And you can see our high mortality. 2005, we shifted our test to where he had planted the nematode resistant variety. And you can see we got a significant reduction in disease. It was still bad, but it was a lot, it was going in the right direction. And then 2006, we shifted to another area, and that had now been 2 years in a nematode resistant variety. And we hardly had any disease at all. So the solution was to get away from what he had been planting and plant something else. But it took 2 years to get the density in the soil to a point where you could grow cotton productively again. I also did a little chemical test in this trial targeting the nematodes. So we used a product called Aldicarb, which targets nematodes, and the blue little squares are the mortality, and here's your mortality scale. And so where we had Aldicarb, we reduced our mortality by 14%, and we increased our yield, which is this bar, by 20%. So by taking the nematode or reducing the nematode with Aldicarb, we did impact the disease to some extent. So this was interesting. This was very interesting, but it wasn't proof. So Jason Woodward was a professor here at Texas Tech. He came around 2006, and he was able to recruit a student, Shilpi Chawla, who came from India, and she was an expert in Fusarium. She had excellent training from India, specifically in Fusarium. So we wanted to set up an experiment to test and try to get a better idea of the impact that the rotation or the lack of rotation with a real susceptible variety, how does that affect the counts of the Fusarium in the soil? And so we had what we called microplots that were put out at the Quaker farm. You excavate a big hole, you put in these structures, and then you fill in the soil with, in this case, from a field that had Fusarium wilt. And then I grew nematodes in the greenhouse, and they added the nematodes in the soil so that an even amount of nematodes was in each plot. We used 2 varieties, Stoneville 4554 B2RF, which was normal susceptible to root knot, but not super susceptible to Fusarium wilt. And we used FiberMax 9058 Flex, which had a pretty high susceptibility to both root knot and Fusarium wilt. And then Shilpi measured the FOV density over time and the wilt incidence each year. And so it was continuous with these 2 varieties, and then several rotations with these 2 varieties. And this is just to give you an idea what a microplot. I don't have a picture for their work at Quaker Farm, but this is an idea of what different microplots may look like. All right, so here's Shilpi's counts for just the Stoneville and the FiberMax variety. I'm not going to complicate it with the rotations right now. So the test started in April of 2008, and there was over 400,000 colony forming units per cc soil, cubic centimeter soil, and it was pretty similar at the start of the experiment for both varieties. 1 year later, we actually had fewer counts behind the Stoneville variety, about a similar level behind the FiberMax. But by the middle of the summer, you can see the FiberMax variety, the numbers were just shooting up. The Stoneville were not changing a whole lot. By the end of the season, you were even a little bit higher with the FiberMax. By spring of the next year, because they would take the plants and they would cut them up and let them sit in the soil over the winter. They were allowing that material to go back in the soil. So by the next spring, we had really maxed out our FiberMax counts, and that maintained through the rest of the year. So you can see using the same really susceptible variety over and over again was far different dynamics than using a normal susceptible variety. Super susceptible versus normal susceptible. Now, these are the wilt counts for the 3 years. In this case, I am going to show the rotations as we get further into time. So we start in 2008. The green is where you had the Stoneville. The red is where you had the FiberMax. So at the end of the first year, and your counts aren't that high the first year, you have significantly higher wilt incidence with your FiberMax than you do with your Stoneville variety. We moved to 2009. So your green is 2 years in a row of Stoneville. Your red is 2 years in a row of FiberMax. Okay, and you can see you have your highest wilt levels where you're 2 years in a row with FiberMax. The blue bar is where you were Stoneville the first year, but FiberMax the second. Okay, it's nearly as bad as 2 years of FiberMax. Your black bar is where you were FiberMax the first year, but Stoneville the second. And you've about doubled from the Stoneville 2 year in a row, but you're still pretty low overall. And then 2010, so here's your green bar, Stoneville 3 years in a row. Here's your FiberMax 3 years in a row. And what are we? We're at 70% incidence of wilt. This light green bar was Stoneville for 2 years and then FiberMax for the third year. Okay, and so you're not that bad with your first 2 years being Stoneville. Just 1 year of the FiberMax wasn't that bad. But this tan one was Stoneville and then 2 years FiberMax. That's a lot worse. And then your yellow bar was 2 years FiberMax, but your third year was Stoneville, and that was actually pretty good. And then this green bar was FiberMax the first year, 2 years of Stoneville, and that, you know, was very little wilt. So you get a feel of how much you can affect the dynamics by using different cultivars. And so this is part of when we're talking with farmers about their strategies. Maybe they prefer the FiberMax varieties. The statistics on the planting acres would suggest they do. And so this is a way for them to grow FiberMax some of the time, but not too many years in a row, right? They may have to go with a variety they're a little bit less happy with in order to keep the populations managed. All right, let's talk about resistance to Fusarium and nematodes. This is some work that Ray Shepard, produced in 1986. In 1974, he developed this variety called Auburn 623 RNR, and it was more resistant to root knot nematode than anything we had ever seen at that point. And it had a very low incidence of Fusarium wilt. So on this graph, the RK number is the reproduction in a greenhouse trial. So you inoculate root knot, you go through it, let it go through a cycle in the greenhouse, and then you count the eggs of root knot at the end of the test. And you take this number and multiply it by 1,000, and that would give you the eggs per plant. So this highly resistant one had less than 1,000 eggs per plant. Cleve Wilt had 12,000 eggs per plant. Coker 100 had 73,000 eggs. Stoneville 213 had 96,000. Dept had 144,000, and the highly susceptible Rowden had 148,000 eggs, okay? And you see the incidence of wilt here on your axis. And so, yeah, there's a pretty good relationship. The more resistance you have to root knot nematode, the less wilt you have, right? It kind of makes sense. The one outlier, and there actually were 2 varieties. This was a test with 18 lines. I just took a subset, but there were 2 outliers, and I showed one of them, Dept 277. This has complete susceptibility to root knot nematode, but it does have some Fusarium wilt resistance. And so there are apparently some Fusarium wilt genes that are separate and not captured within the resistance that the root knot nematode would bring. But nematologists such as myself working in cotton took this kind of information and said, well, all we need is resistance to root knot nematode, and we don't have to worry about Fusarium wilt. And so that's the target we went after, the root knot nematode resistance. So this gives you a little bit of a history of Fusarium wilt resistant sources, and it's messy. So if you go all the way back to the 1800s and you go back to founder germplasm, you had 3 sort of lines of founder germplasm, the Jackson limbless, Petit Gulf, and Weiss. That's where Fusarium wilt resistance came from, those 3 sources. Weiss had a commercial variety, Wild, in 1928 that was planted on some acres that had Fusarium wilt. The Petit Gulf had Cook 307-6 and then Coker Cleve Wilt, and those were good sources of Fusarium wilt resistance. The Cleve Wilt was also in that previous slide. Whoops, sorry. Here was Cleve Wilt. Okay, so it had some pretty good resistance as well. Dillon was released in 1900 with some resistance, but Dixie Triumph was actually a pretty good variety for that time, and it was actually planted commercially on some acres. But for whatever reason, all the commercial varieties that we have today came from that middle branch. Okay, so Cleve Wilt was crossed with Coker 100, which was an excellent variety for the time, but it was susceptible to Fusarium Wilt. So it was crossed with Coker 100 and made Coker 100 Wilt. That was a commercial variety that got some acres. And that was crossed with Cook 307-6, which also had Fusarium Wilt resistance and developed into Auburn 56. So there was a time where Auburn 56 was considered one of the best Fusarium Wilt resistant varieties, but agronomically, yeah, not so good. So back over in Louisiana, the USDA there, they felt like Bayou, which had come from Cleve Wilt by Delta Pine 15, they felt like Bayou was a little better agronomy or agronomic characteristics and equal Fusarium Wilt. So they kept working with it, developed Delta Pine, crossed it with Delta Pine 16, developed this one, crossed with that, and they came up with Stoneville LA 887. Well, that variety was the first commercial variety that I saw. It came out in the late 1990s. And the fiber quality was dreadful and it would just fall off the plant. But if you could catch it in time, it actually out-yielded the commercial varieties out here in a root knot field. And so I was kind of impressed. And this became sort of the parent of a lot of the Stoneville resistant varieties that were developed after that. The other thing is we already talked about Auburn 623 and how highly resistant it was. Well, it was developed by Cleve Wilt, a selection in Cleve Wilt times Mexican Wild Jack Jones. And they both had root knot resistance, different genes though. And so this became what I think the Delta Pine and the Photogen folks, all the resistant varieties that they developed after that, they sort of started with this as their source. And that had 2 gene resistance. Stoneville LA 887 just had the Cleve Wilt, so it only had 1 of the 2 genes. But the Auburn 623, that had both. That had 2 genes. All right, let's look at the nematode life cycle. So in the spring, you have the second stage juveniles, and they can move through the soil, and they can infect a root. And when they infect a root, they have to develop a specialized feeding site. They have to develop a special relationship. And when they do successfully, then the root starts to swell. The nematodes are putting spit into the plant. It's increasing these changes. And then they develop, the nematode swells, the root develops a gall. And then the nematode goes through several molts in the root, those J3 and J4 stages, to a female. And then the female produces eggs, the eggs hatch, and you go through multiple generations in a season. So I want you to think about this, and we're going to look at some data to try to explore. We have 2 root knot resistant genes that we know of. We have 2 in cotton. And we're going to look at them, and they came out of that Auburn, and we're going to look at them and what they may do to this life cycle. So this was a study that De Silva et al. Published in 2019. They took a line that was developed from the Auburn, and it was called M120. It had the 2 resistant genes. One was on chromosome 11, and we'll call it RK1, and one was on chromosome 14, and we'll call it RK2. Anyway, they decided to put 1 gene into an isoline, and then the other gene into another isoline. Isoline meaning the varieties were basically the same, other than those 2 genes. And then M120 was your resistant parent that had both genes. And then Coker was your susceptible parent that didn't have any of the genes. And so the first thing we're going to look at is the galls, the number of galls per plant, and that's the bars. So the Coker had about 300 galls per plant. The M120 had something like 75 galls per plant. The RK1 gene on chromosome 11 was pretty similar to the M120, okay? So galls form initially when the J2 get in the root and develop that feeding site. So that would suggest RK1 gene goes after the life cycle really early in the process. The chromosome 14 gene, it had lots of galls. So clearly that's not affecting the galling. It's not affecting that part of the nematode life cycle. The red line are the eggs per plant. And so after 40 days, they measured the eggs, one life cycle, they measured the eggs, and they had about 170,000 eggs in your susceptible parent and about 20,000 eggs in your resistant parent. But both of the isolines had about 40,000 eggs. And so this was resistant. I mean, it greatly reduced the eggs, even though there were lots of galls. So clearly it's affecting maybe how long it takes to become an adult or something about the production of the eggs. There's speculation as in terms of what it's affecting, but it's clearly farther along in the life cycle. So when you add the two genes together, they're really powerful because they're both affecting different things. And so there's really not a lot of overlap. They are affecting different parts of the life cycle, and they work really well together. Now, this messy slide is all the modern root knot nematode resistant varieties. Some varieties have the 2-gene resistance, the highest level of resistance, and they have it in every plant. Okay, they have it homogeneous in the population. Some varieties have 1 gene, and they have it in every plant. They have it homogeneous. Some have 1 gene, and they only have it in a fraction of the population. So you have a lot of, when you say resistance, in some cases you're 95% reduction of nematodes, and in some cases you may be down to like 25% reduction of nematodes. The range of resistance is quite different depending on the variety. And I've colored different companies' varieties, different colors. But you can see we're continuing to add varieties over time. Now, what I've highlighted in red are the years, the varieties that came out in those years, because all of a sudden we started getting calls again, and there was some catastrophic Fusarium wilt showing up. In this case, it was showing up on these nematode-resistant varieties. It was worse than a susceptible variety, okay? Worse than a susceptible variety. So here we go with excellent resistant material. The companies are using markers now. Delta Pine and Phytogen, they're using markers. They can target the root knot genes very precisely. They're using markers. And that's where we're getting calls that we've got catastrophic Fusarium wilt showing up again. It made no sense. They're highly resistant varieties. So we started doing variety tests. We went to one of these sites that had catastrophic losses, and we put in a variety test. And Dr. Monclova Santana, who was here for a couple years, she came in 2019, and we worked together the first year, and she sort of took charge after that. And we ran this project for 3 years. So this is that field that you saw in that picture. So we'll start with the mortality. So again, mortality is what came up originally and then started to die. Now, the red bar is susceptible, nematode susceptible varieties. This second bar is Stoneville 4946 GLB2. It's a variety from Stoneville, and it's what we recommended in fields that had Fusarium wilt problems. It's not the most nematode resistant variety out there. It's sort of intermediate, but it's a good Fusarium wilt variety. We have this Stoneville 5600 B2XF. This was actually, it wasn't developed by Stoneville. I think they probably bought it from Monsanto back in the day. But anyway, so it had a different genetics than the Stoneville 4946. It didn't come along the same way. This one would have come along through that Stoneville LA887. If you remember a couple charts back where we talked about how the varieties came about, this one came from that Stoneville LA887. It did not come from the Auburn line. It came from that other source that would have come just from the cleave wilt, okay? And it was developed not with marker technology. This one is old enough that it was developed by going to the field and making selections, going to a nematode Fusarium wilt field and making selections. That's how this was developed. The Delta Pine resistant varieties were developed by molecular technology, molecular markers, okay? And probably this one was as well, even though we don't have what its history is because it was licensed. It wasn't developed by Stoneville. So our mortality data was confusing. We did have a lot of mortality, over 40% in our susceptible varieties. So it was a bad field, but we didn't get very clean separation with our different varieties, very noisy data. I look at the root knot counts and they look good. They look like they're supposed to. The susceptible variety had a lot higher counts than your nematode resistant varieties. And this one has a weaker source of resistance. So I would expect there to be a little more reproduction in there than these two varieties. So everything makes sense from a nematode standpoint, but the mortality data is very confusing. And the farmer did turn off the pivot because the Fusarium was so bad. And so the yields are low, but you can see that the Stoneville 4946 was the highest yielding variety in that location. This is a drone image that we took of the field. And hopefully you can see sort of the bare areas. You can see our plots, and these are the alleyways at the end of our plots. And hopefully you can see the sort of the bare areas where the Fusarium wilt was the worst. Well, this is the problem. How on earth can you do a variety test when your disease is that variable? I mean, it was a bad field, but how can you do a variety test? Because you've got some varieties where there's almost no disease, and then you've got others where everything's being wiped out. So that was the problem. What are we going to do? We can't, I cannot tell you definitively if this is a good Fusarium wilt variety or a bad one. What are you going to do? Students, what are you going to do in this situation? Any ideas? You're going to go to the greenhouse. Whoops. Okay, well, we'll go beyond that. So you're going to go to the greenhouse and you're going to test varieties in the greenhouse where you can control your inoculum levels. Okay, so this is 3 different methods that you could use that have been published for greenhouse testing. And we chose to use the middle. And so what we did is we planted seed in soil that had root knot nematode, and we let it grow for 2 weeks or so. And then we would carefully remove the plants from the soil, very sandy soil, wash the roots off, and then we would dip those roots in a spore solution of the fungus, okay? We'd have a certain density, and we would dip all the plants into that, and then we would carefully transplant each plant into its own cup after that. And then we'd rate them. And the person who did this project was Jessica Dautre, and she was a master's student here at Texas Tech. And so here's some symptoms that she would rate by. So 0 is no disease symptoms. 1 is where one cotyledon is wilting. 2 is where both cotyledons are wilting. 3 is where it progresses to sort of a yellow color. 4 is where it's starting to dry up and die, but there's still a little bit of green tissue. And then 5 is a completely dead plant. And so Jesse would rate the plants over time and then use what's called an area under disease progress curve to combine the data together over time to give you a visual foliar rating. And then she would also, at the end of the experiment, she would look at the roots and rate those for percent necrosis, and then cut the stem and rate the stem for percent necrosis. And here's just some pictures of her stuff where you can see the vascular discoloration in the root. So she's trying to estimate the percent of vascular discoloration in the roots. And then there's one of the stem where you can see it's close to 100% vascular discoloration. So here's some of her combined analysis from a couple trials. The red varieties are susceptible to root knot nematode. The yellow varieties are resistant to root knot nematode, but just partial resistance. They either have just 1 gene, not 2, or they have 1 gene that's even not in every plant. That's just in part of the population. The green is a 2-gene resistant variety. Okay, that has both genes in every plant. So the highest level of resistance. So the bars are this foliar symptom, area under disease progress curve. So the higher the bar, the more disease, the more foliar symptoms. So you can see the highest level of disease, which is your A's, were in Delta Pine 1646 and NextGen 4936. Those are susceptible varieties. They're in Phytogen 480, which is your highly resistant variety, and it's in Delta Pine 1747 NR B3XF. And these varieties are some of the varieties that were associated with the catastrophic Fusarium wilt field problems that we were getting calls on, okay? So clearly, nematode resistance is no longer equal to Fusarium wilt resistance. And I think these varieties were developed with markers, excellent markers for the nematode, the two nematode resistant genes. So somehow the Fusarium wilt may have been in close proximity to those genes, but somehow by using the markers, they lost the Fusarium wilt resistance. And I don't know why they're even more susceptible in some cases than susceptible varieties, but this Jessica's work proved that nematode resistance is no longer connected to Fusarium wilt resistance. You cannot assume a nematode resistant variety is highly resistant to Fusarium wilt. You can't make that assumption, especially if you're using markers and not making some selections in a field that has both pathogens. Now, the black line was the root necrosis ratings, and the blue line is your stem necrosis ratings. And you can see that they trend pretty well with the foliar symptoms. And this one variety that's the best, that's Stoneville 4946 GLB2. Okay, so which was not developed with marker technology. Okay, was done the old-fashioned way. Progress sometimes isn't always progress. We have really good nematode resistance now, but ouch. All right, so I'm going to shift gears. And so all of that were root knot nematode race 1 and 2 of Fusarium wilt, okay? Now we're going to talk just briefly about race 3 of Fusarium wilt. So I didn't think we had much race 3 out here. I certainly didn't think it was a problem. We have been working on some molecular assays to be able to identify FOV race by directly testing the DNA from roots. So you extract the fungal DNA, and Jennifer Chagoya is a graduate student here at Texas Tech, and she's been doing this work. And they started it with Dr. Monclova Santana back in 2019 or 2020. But anyway, the idea was normally you have to do isolations, you have to pull the fungi from the roots, and then you would have to do some sequencing of those fungi to see if you have FOV as opposed to another type of Fusarium, and to determine the race that you have. And it takes a lot of time, and it's a lot of effort. And so it's not something that would be done routinely. So the idea was to develop an assay that would be rapid, just like one day. And even better if you didn't have to do the isolation step, if you could directly assay the roots. And so last year, Jennifer Chagoya was testing her protocol, her procedure, on a wide array of samples that came from 67 fields across the Southern High Plains. Some were healthy fields, some were disease fields. Only one of those 67 was a known Fusarium wilt field. Well, the results were a bit astonishing. First of all, her assay worked really well, and I expect that she'll give a seminar in this later in the year as she finishes her Ph.D. But one of the astonishing things to me was how much race 3. So we were assaying roots. We weren't taking it from the stem, which is where we find race 1 and 2. We were just assaying the roots. Well, a high proportion of the roots that came from diseased plants, not wilt plants, but roots that had necrosis, fields that didn't grow right. The farmers, oh, what's going on in my field, right? Well, a high proportion of those had FOV3 in it, race 3. So that kind of opens up a new line of research. We need to know if race 3 is, now you don't always find race 3 by itself. Sometimes it's with race 1 or race 2. So it can be a mixture, right? And there are other Fusariums that are in the roots as well as sometimes other pathogens. But there was such a high proportion of FOV race 3 in these diseased roots and such a low proportion in the healthy roots that's an area that we'll have to get active in. Now that we've had this eye-opening experience, we need to investigate this further. We need to try to determine if race 3 is actually causing root disease problems in this region. So we'll just have to see how that progresses. We have a fair number of isolates that we can start testing. And then the big elephant in the room is FOV race 4. So this was discovered in the El Paso region in 2017. It was found in California back around 2002, and probably been there for several years before that. And it wiped out Pima production in California and until they had a resistant variety, which fortunately they found one pretty quickly, but it was Phytogen 800. But until they found that resistant variety, they could not grow Pima cotton when race 4 was present. And it got bad enough eventually that the upland types, which is what we typically grow here, the upland types didn't grow very well on it either. And so we've always kind of kept our eye on that California situation, and it stayed in California for a lot of years. But in 2017, a Pima breeder who was growing cotton nurseries in the El Paso area alerted us that it was there. And he actually had spotted it back in 2012, which is another story. But so it's been out in the El Paso area since at least 2012. And we didn't know about it. So seed has been moved around, right, since 2000 and whatever. Now I think seed production, seed movement has stopped since it was discovered. I mean, the company stopped using that as nursery sites, right? But that's a lot of potential for movement of FOV4 around the state. And so now that we have this assay, we're going to start testing big time. If it's found out here in the Southern High Plains, it will be a little bit earth-shattering for some years because of its seed spread. We produce about 50% of the U.S. cotton planting seed here in Texas, about 50%. So can you imagine what all the other states are going to say, right, if it's found in a region that is producing half of the U.S. planting seed? So it'll be disruptive. On the good note, there's been a tremendous amount of work done looking for resistance in upland cotton. And really the leader of the pack is Mauricio Loa here at the USDA. He started working with it in California. And in a couple of years ago, he released a paper where he had 17, I think most of them upland cotton lines that he released that have some degree of resistance. And he's found a source of resistance, which is really, really quite excellent. And so it needs a lot of work in terms of crossing with better material, but there's a good source of upland resistance. There are markers that have been developed for that source now out of New Mexico State and published. And so really the table's there. If the companies want to start making crosses with that material, it would be helpful so that when it is found elsewhere, we will have, resistant varieties close to where they could be sold. Phytogen this past winter, and I talked to grower meetings in the winter, and Phytogen announced at the grower meetings this past winter that they are bringing one forward to marketplace. Now, it may be 4 years out or something. It's still a little ways out, but they actually have one that they're bringing to the marketplace pretty soon. So that was reassuring to hear that. But majority of our varieties are quite susceptible to FOV race 4 at this time. And you saw what happened from some of the early data sets, what happens when you plant highly susceptible varieties a couple years in a row. That fungal density is really going to blow up. And that fungus does not need root knot nematode. FOV4 can kill cotton without root knot nematode. So it would potentially open up our entire region to having problems with it. Okay, so I think that's just revisiting what I talked about. The only other point I'll make is we still have problems with race 1 and 2 out here. And it sure would be nice if we could have markers developed for that source of resistance, because we've lost it because we're using the root knot markers and we're not using a Fusarium marker for the source of resistance that does exist. And so it would be a really nice project, I think, to get molecular markers to probably that cleave wilt source of resistance that's separate probably from the root knot source. All right, so are there any questions?
What actually driving the resistance of those like a highly resistant variety?
There does not seem to be any understanding of the mechanism of the resistance. And you got to remember too, we didn't even know it was separate from the root knot resistance until, it started affecting the marker-assisted selected resistant varieties. Now for FOV4, there's been a nice paper that was published, some nice work out of Yi Zhu out of New Mexico State, has a couple papers where she's shown with Pima and with upland that you get far fewer mycelia colonizing the roots with the resistant as opposed to susceptible. And so it's affecting the ability of the fungus to colonize the roots. It's affecting their ability to get into the center of the root. It's affecting its ability to spread in the plant. So it seems like all stages of the FOV4, all stages are being affected by the resistant gene when it occurs. And she did a lot of photographs and followed the actual growth in the root all the way through the cycle. And it was very impressive. So in terms of mechanistically, I don't exactly know what's happening, but it's whatever it is, it's a wonderful source of resistance because it seems to be affecting the fungus at each step of the way. For the FOV race 1 and 2 sources of resistance, no idea. That just wasn't worked on because everyone assumed it was the root knot resistance that was the reason that you didn't have Fusarium wilt. And so it wasn't till 2018 or 19 that we started to realize, apparently there was separate genes involved with the Fusarium wilt resistance besides the root knot, and we've disconnected them by using the markers for root knot. But great work for a student if they want to do it. Yes, sir.
Was seen in 2017, right?
So the confirmation was in 2017, but that breeder who brought it to our attention in 2017 actually found it in 2012 out there, but he started developing nurseries. And it takes 4 years or so for this breeder to develop a good FOV4 nursery. So he waited until 4 years later. And then, whoa, we've got it in Texas. So out there they furrow water their Pima cotton. It's a lot of Pima cotton. They furrow water. They get the water from the Rio Grande. So they really flood irrigate and, put a lot of water on for that Pima cotton because it's much higher value. And it's always dry in El Paso. I mean, a wet year is maybe 6 inches, right? So that doesn't affect the lack of rain. Maybe the temperature will have some impact, and I don't remember what the temperature was in 2017. And really it was 2016 when it got the growers' attention big time. If you look at satellite images from 2016, it was terribly damaging. We didn't have a plant pathologist in that part of the state. And so unfortunately, it didn't come to our attention until 2017. And then Jason Woodward went out there and it was like, oh my gosh, this looks like race 4, because he had been paying attention to the California situation. And sure enough, collected a lot of material and it was sent to some different people to do the molecular tests, and it was confirmed. Yeah. And then New Mexico State confirmed it in New Mexico across the border. Basically, you're very close to the border there, and they found it that next year in New Mexico State or New Mexico. Yeah. So, but it hasn't been found anywhere else yet.
Regarding the fungin resistance, the Fusarium, the very catastrophic disease, as well as nematode helps to penetrate the in the plant cell. But is there any field difference or regional difference of the fungin resistant variety in US in aspect of the Fusarium disease severity?
Okay, is there any difference? Say that again.
it means a field difference. I
A,
mean,
a field difference.
field difference across the.
That's a good question. So I would have said no. I would have said no, but this year at the Cotton Belt-wide meeting, for the first time, there was some comments from other parts of the state or of the country where people were starting to wonder if this company's 2-gene resistant varieties held the populations lower and this company's maybe let a little more reproduction. And the main players are Delta Pine and Phytogen, okay? And so one company, there was some rumbling from some of the nematologists that maybe this company's 2-gene one was a little better than this company's 2-gene one. I have not seen that out here at all. There's some recent work with the soybean cyst nematode that has shown that some sources of resistance were better for a number of years if you had a higher copy number of the resistant gene. And then this other that might have had the same gene, but it had a much lower copy number, and that affected how good the resistant gene was. And so, and this is, I mean, this is just since January that I heard this, and I think there'll be some interest in maybe trying to look into whether there may be some copy number differences between the different companies' varieties. But myself, I have not measured, and I do a lot of nematode counts, and I have not seen one variety's counts be, with the 2-gene resistance, be any different than the other companies' varieties. I haven't seen it at all. So we do have lower populations out here than areas where it rains more. So a damaging level here may be half of what a damaging level in Louisiana or Georgia or someplace would be. And we have more water stress, and so we need fewer nematodes to cause damage, but they also don't build up as high because there's restrictions. They have to have soil moisture to move and infect roots, and that can be real limiting here. So. Right, yeah. So water stress on the plant, we need fewer nematodes to cause the same damage that they need more nematodes in Georgia, but they have more nematodes in Georgia because it rains. So, yeah. Anything else?
Okay, thank
Good.
you, Doctor.